Hello students, this is the second video of the chapter Life Processes, where we will be discussing nutrition in plants, that is autotrophic mode of nutrition. We have already discussed the introduction where I told you the various life processes, that is nutrition, respiration, transportation, excretion, etc. So when we take up the life process nutrition, we have to discuss both autotrophic and heterotrophic mode of nutrition. So first of all, what is this nutrition? Taking in the source of energy, that is food, from outside to inside. This process is known as nutrition. There are two modes of nutrition, that is autotrophic and heterotrophic mode of nutrition. What is autotrophic mode of nutrition? The process in which green plants and blue-green algae, they prepare their own food. Or in other words, we can say preparation of complex organic substances from simple inorganic substances. Now what is this complex organic substance? Glucose. And the simple inorganic substances are water, carbon dioxide, minerals, etc. And the organisms which follow this mode are known as autotrophs. Examples are green plants and blue-green algae. Heterotrophic mode of nutrition is a mode in which organisms depend on other organisms for their nutritional needs. Or else we can say breaking down of complex organic substance into simple inorganic substances. Now what are these complex organic substances? Carbohydrates, proteins, fats, etc. While it is broken down into simple substances such as carbon dioxide, water, and of course energy is released. And the organisms which follow this mode are known as heterotrophs. Here let us focus on autotrophic mode of nutrition. Autotrophic mode of nutrition means the method that is followed by green plants and blue-green algae, and that is known as photosynthesis. Now photo means light and synthesis means preparation. So using light preparing food. So what is the definition? Formation of glucose using carbon dioxide, water and minerals in presence of sunlight and chlorophyll. Now this glucose is then broken down, energy is released, and it is this energy is utilized for the various functions of the plant body. But all of the glucose that is produced is not required immediately. Part of it is stored. So in which form is it stored? It is stored in the form of starch. So the internal energy reserve in plants is known as starch, while the internal energy reserve in plants, in animals, is known as glycogen. Now let us see what is the chemical equation for photosynthesis. 6 molecules of carbon dioxide plus 12 molecules of water in presence of sunlight and chlorophyll gives 1 molecule of glucose, 6 molecules of oxygen and 6 molecules of water. Or else we can say 6 CO2 plus 12 H2O in presence of chlorophyll and sunlight gives 1 molecule of C6H12 plus 6 molecules of O2 plus 6 molecules of H2O. So this is the chemical equation, and that too it is the balanced chemical equation. Now what are the 3 steps involved in photosynthesis in normal plants? First step, taking in of or absorbing solar energy by chlorophyll. This solar energy or light energy is then converted to chemical energy. and splitting of water into hydrogen and water molecules. Third step is reduction of carbon dioxide to carbohydrate. Once again, I repeat, light energy is absorbed by chlorophyll. This light energy is converted to chemical energy. Then splitting of water molecule into oxygen and hydrogen takes place. Then reduction of carbon dioxide to carbohydrate. But in desert plants, this is not the way it happens, because desert plants, the availability of water is less. So the stomata remains closed during the daytime. And you know the two functions of stomata are exchange of gases and transpiration. So if stomata remains closed, both transpiration and exchange of gases cannot take place. That means carbon dioxide cannot be absorbed during the daytime. So stomata opens only at night. So carbon dioxide is absorbed during the night. Once it is absorbed, an intermediate is formed, and this intermediate is acted upon by the energy that was captured during the daytime. So that is how photosynthesis happens in desert plants. Once again I repeat, carbon dioxide is taken in during the night, intermediate is formed, and this intermediate is acted upon by the energy that was captured during the daytime. Now, let us discuss about stomata. Stomata are tiny pores found on leaf surfaces. They have two functions, exchange of gases and transpiration. By exchange of gases, I mean taking enough carbon dioxide and giving out oxygen during photosynthesis. And during respiration, taking enough oxygen and giving out carbon dioxide. Transpiration is the loss of excess amount of water in the form of water vapor. Now let us discuss the structure of a stomata. Stomata has a tiny pore in the center, that is known as the stomatal pore. This is guarded by two bean-shaped structures known as guard cells. The guard cell, the inner wall, that is the concave wall, is thicker than the convex wall. The guard cells contain nucleus and chloroplast, and this is surrounded by epidermal cells. So the important parts are stomatal pore, guard cells, and nucleus and chloroplast that are present in the guard cells, and the epidermal cells surrounding it. Next, what is the function of this guard cell? Guard cells, they help to regulate the opening and closing of the stomatal pore. Now, what is the mechanism which causes this opening and closing? When water enters through osmosis into the guard cells from the surrounding cells, the guard cells swell or become turgid and causes the opening of the stomatal pore. When water leaves the guard cells through diffusion, through osmosis, the guard cells, they shrink. Let us discuss the difference between guard cells in dicot plants and monocot plants. Guard cells in dicot plants are kidney-shaped or bean-shaped structures, while guard cells in the monocot plants are dumbbell-shaped. Not only that, in dicot plants, the stomata are found only in the lower epidermis, while in monocot plants, stomata are found both in upper and lower surface. That is, upper epidermis and the lower epidermis. So these are the differences with regard to stomata in case of dicot and monocot plants. Next, let us see what is the function of guard cells. Function of guard cell is to regulate the opening and closing of stomata. What is the mechanism of opening and closing? When water from the surrounding cells enter the guard cells swell, which causes the opening of the pore. And when the water leaves the guard cells shrink, causing the closing of the stomata. So this is how the opening and closing of stomata of the stomatal pore take place, and guard cells are responsible for the regulation of opening and closing of the stomata. Next, let us see a few experiments, that is experiment 6.1, where we need to prove that chlorophyll is necessary for photosynthesis. For this, we have to take a potted plant with variegated leaf. By variegated leaf, I mean colored leaf. For example, coleus, money plant, crotons, all they have variegated leaves. We have to keep this potted plant in darkness for 3 days so as to discharge the plant. Then we need to keep this plant in sunlight for 6 hours so that starch is freshly prepared. Then we can pluck one leaf from this plant and take a tracing paper and trace out the various patterns there and mark the green parts clearly and keep it so as to we can compare at last. Next thing is performance of the starch test. This leaf which we have plucked, we have to boil it in a beaker containing alcohol. But this beaker containing alcohol cannot be kept directly on the flame. It has to be kept in a water bath. What is the reason? Because alcohol is highly flammable. So this beaker containing alcohol has to be kept in a water bath and then placed on the flame. So when we boil this leaf in alcohol, it becomes colorless, and you can see that color oozes out, and the alcohol, it becomes green in color. This leaf, this colorless leaf, then has to be taken out. It is somewhat brittle, so we need to rinse it in water so that the tissue softens. Then we have to take a dropper and pour few drops of iodine over the leaf. We will observe that those parts which are green in color, which they become blue-black, and the other parts, that is other parts which were colored yellow or red, they do not turn blue, they remain colorless. You can compare it with the diagram or the drawing which you have made it in the this paper, then you will understand which parts are green and which were non-green, and you will observe that the parts where you had marked green exactly has turned blue. So what does this experiment prove? Chlorophyll is necessary for photosynthesis. Next, let's see activity 6.2, where we have to prove that carbon dioxide is necessary for photosynthesis. For this, we need to take 2 potted plants which are similar in size. Again, keep it in darkness for 3 days to discharge them. Then take them, and before placing it in sunlight, we have to do certain preparations. We have to label both the pots A and B and place these on glass plates. Now on the glass plate of pot A, we have to place a petri dish containing potassium hydroxide, and on the pot B, glass plate of pot B, we will not place this chemical. After placing the chemical, then we need to take 2 glass bell jars, and place it over both the pots and seal the space between the bell jar and the glass plates with Vaseline. This we have to do for both pot A and B. Why is this done? This is done to make the experimental setup airtight, or else carbon dioxide may get in from the atmosphere through the space. Now we have to keep this experimental setup A and B in sunlight for 6 hours. After keeping it in sunlight for 6 hours, we have to pluck one leaf each from both the pots and perform the starch test. That is boiling in alcohol, rinsing in water, and then pouring a few drops of iodine. When we do that, we see the leaf that we pluck from A remains colorless, while the leaf that we got from B becomes blue-black. This proves that carbon dioxide is necessary for photosynthesis. So what happened in the experimental setup A? The chemical potassium hydroxide, it absorbed all the carbon dioxide that was present inside the bell jar. So carbon dioxide was not available for the plant. So photosynthesis did not take place. Therefore starch was not prepared. So we have seen that chlorophyll is necessary for photosynthesis. Carbon dioxide is necessary for photosynthesis. Let us take the third experiment. In your textbook it is written, can we design an experiment to demonstrate sunlight is essential for photosynthesis? For this we have to take a potted plant. Again, keep it in darkness for 3 days to discharge it. But before keeping it in sunlight, we have to select one leaf and place a black strip of paper. We have to clip a black strip of paper on one of the leaves, a small strip in the center. Then we have to place it in sunlight for 6 hours. This leaf we have to pluck. Remove the black strip and boil it in alcohol. Then rinse in water and perform the starch test. What shall we observe? We will see that the portion where we had placed this black strip of paper, it remains colorless, and the regions which were exposed becomes blue-black. So what does this prove? Sunlight is necessary for photosynthesis. Now, I told you water and minerals are absorbed by the roots. Now, what are the important minerals that are needed by the plants? They are nitrogen, phosphorus, iron, magnesium. Among this, nitrogen is the most important because it is essential for making proteins and other components. Now, how is this nitrogen absorbed? In which form it is absorbed? It is absorbed in the form of nitrates and nitrites in inorganic forms, and in organic form it is absorbed in the form of nitrogen in organic form is absorbed by the nitrogen fixing bacteria, that is rhizobium. So here, if I read out that portion for you, see, it is taken up in the form of inorganic nitrates and nitrites, or it is taken as organic compounds which are prepared by whom? Which are prepared by nitrogen fixing bacteria, that is rhizobium. So with that, we have concluded this topic of autotrophic mode of nutrition. Thank you.